Um, Hello, everyone. I'm Chris Williams. I'm the Chief Business Officer at Autolus. I'm delighted to share our company updates at Meeting on the Med this year. Let me get the slide clicker working. Here are our disclaimers. Just a little introduction to Autolus for those of you who are not familiar with the company. We're an autologous T-cell therapy company focused on the delivery of life-changing treatments for cancer and autoimmune diseases. Our lead candidate, Obel, is in late-stage development for relapsed refractory adult ALL, and our BLA is currently being reviewed by the FDA. Beyond ALL, Obel is being developed in earlier phase clinical trials for non-Hodgkin's lymphomas, CLL, and most recently we initiated the Carlisle phase 1 study in patients with lupus. The development and planned commercialization of Obel is underpinned by our state-of-the-art manufacturing facility, which is located in the UK, and you can see the pictures of the outside and one of our clean rooms in the facility on the right-hand side of the slide. Beyond Obel, we're developing a pipeline with broad applicability in cancers with limited treatment options. And Autolus is underpinned by a strong technology foundation developed by Martin Poulet, our CSO and academic founder. And we're working in partnership with multiple companies to broadly exploit this potential. Our next few slides will introduce Obel and where we are with this program. So Obel is a CD19 CAR T-cell therapy with a unique mechanism of action. Most of the approved products in this space are based on the FMC63 binder, which has a very high affinity for the CD19 target. This high affinity interaction can tether the CAR T-cells to their target cells and lead to overactivation and excessive cytokine release. This can cause toxicities in patients and also exhaustion of the CAR T-cells, which potentially impairs the efficacy. The approach we took with Obel was to take a unique binder called the CAT binder, which has a similar on rate to FMC63, but has a very fast off rate and disengages very rapidly. This transient interaction is similar to how a normal T-cell behaves when it engages with a target peptide presented by MHC. So Obel is designed to have a more physiological T-cell interaction with target cells in order to avoid this overactivation, and we hope has the potential to improve efficacy and safety for patients receiving this therapy. Initially, we're developing Obel in adult ALL. These patients are frail and prone to comorbidities, and we believe Obel has the potential to be differentiated from approved therapies in this setting. The FELIX study is our pivotal clinical study in relapsed refractory adult ALL. The study was conducted across 35 clinical sites in the US, UK, and Spain, with product manufactured from the UK. At the ASH meeting at the end of last year, we presented pooled data from the phase 1b and phase 2 cohorts from this study. I'm going to share some of this data on the next few slides. In the bottom panel of this slide, you can see that 83% of the enrolled patients were infused with Obel, and these patients were heavily pretreated adult BLL patients, many of which had received prior bone marrow transplants. Patients who received Obel had high molecular complete remission rates. The patients with morphological disease, which is defined as patients having greater than 5% blasts in their bone marrow, showed a complete response rate of 74%, and in a smaller cohort of patients with less than 5% blasts, all patients went into complete molecular remission. In terms of long-term outcomes, we began to see a trend for long-term outcomes in patients that responded to Obel. The event-free survival in this cohort was at 12 months was 50%, and this is with a median follow-up time of around 16 and a half months. In terms of toxicities, if you look at the left-hand panel of this slide, you can see we saw very low rates of high-grade CRS or ICANS, which is the cell therapy-related neurotox. If you look at the right-hand panel, this shows the data in terms of manufacturing and product delivery. So from a product delivery perspective, our fully enclosed automated manufacturing process delivers very consistent and robust product. And this is despite a very challenging starting material. These patients often have high B-cell blasts within the leukapheresis that we have to manage through manufacturing. The far right-hand panel here shows our turnaround time, and you can see our vein to release time was 22 days, which we're very excited about considering this study was conducted in part through the COVID pandemic with all of those associated complications of logistics. This talks about the next steps for Obel. As I mentioned earlier, we've filed our BLA and it's under review. We have a PDUFA date of November this year. We recently filed the MAA with the EMA, and this is being validated and is being reviewed by the EMA. And most recently, our manufacturing facility was inspected by the MHRA, and we've received a license for commercial manufacturing from those guys. Beyond the regulatory activities, my commercial colleagues who were represented by Chris Van and Dilip Patel at this meeting are focused on multiple work streams to bring Obel to launch preparedness. We're also, as I mentioned before, working on expanding the opportunity beyond Obel in adult ALL. And initially, we are focused on the development of Obel in patients with severe and refractory lupus. We've initiated the phase 1 Carlisle study. Many of you are aware, and if you were in this session earlier, I'm sure this data was discussed, that academic groups of Georg Schett and Andreas Mackerson have shown that autologous CD19 CARs could have transformational benefits for patients with life-threatening autoimmune diseases. We believe Obel is well placed for development in the autoimmune disease setting. We can leverage the safety profile that we see in the oncology indications, which we believe it has the potential to be best in class. We also see very good expansion of Obel compared to other CAR T cells, and we think this will drive the deep remissions required for disease amelioration in autoimmune disease. And then finally, we hope to be able to leverage our oncology data package and the safety data into a more expedited path for development in autoimmune diseases. Beyond Obel, we have a broad pipeline, as I mentioned earlier. I'll just touch on 2 programs where we've had recent updates. Our Auto8 program is in a phase 1 study for patients with refractory and relapsed multiple myeloma. And here we combine a very sensitive BCMA CAR, which is designed to target cells that express very low levels of BCMA, with the CD19 CAR from Obel. And the aim here is to drive durable remissions in this disease. Our initial data presented at ASH shows that Auto8 was very well tolerated in all patients that received the therapy, no dose-limiting tox, very low levels of CRS and no neurotox. And we saw responses in all of the patients we treated. We could continue to treat patients on this study, and we'll look at the data in terms of long-term durability towards the end of this year. We have initiated our first solid tumor program, which is a phase 1 clinical study for a product called Auto6NG in relapsed refractory neuroblastoma, which is a neuroendocrine tumor that affects children. We're working with UCL and Great Ormond Street Hospital on this study. Auto6NG combines a clinically validated GD2 CAR with engineering modules designed at enhancing persistence and activity in the hostile tumor microenvironment you see in solid tumors. Finally, I mentioned some of the strategic collaborations we've entered into to expand the potential of our pipeline. Most notably, and recently, we announced a collaboration with BioNTech, which is aimed at advancing our programs and also their programs through development. The capital raised from the BioNTech transaction will be used to support the launch of Obel and the expansion into new indications. And BioNTech has the option to leverage our manufacturing and commercial capabilities for their BNT211 program, which is in early clinical development for Claudin-6 positive solid tumors. We also have options to collaborate with BioNTech on our Auto122 program and the Auto6NG program that I introduced to you a moment ago. And I think that's the end of my presentation. Thank you very much for listening.